Thank you very much. It's an honor to be here. Having been one of the speakers at the 1987 symposium, it's good to be back and to be talking about the same creatures. I'm here today on behalf of the staff, the students, and our collaborators with the Chicago Zoological Society's Sarasota Dolphin Research Program. And I'd like to speak with you today about how Sarasota Bay's bottlenose dolphins can serve as indicators of ecosystem health. So what might our fin neighbors be able to tell us about the health of our coastal ecosystem? First of all, they're large mammals with physiology similar to ours. They breathe the same air, they swim in the same waters, they eat the same fish, and they share our backyard through decades-long lives. In contrast, they have greater exposure to the waters than we do. They breathe air without the benefits of our nasal filtering structures, and they eat a lot more fish than we do. So they're likely to experience changes to our shared coastal ecosystem before we do. We operate the world's longest running study of a wild dolphin population. Over the years, our approach has been to do longitudinal research, monitoring the biology, the behavior, and the ecology of individual dolphins and their ecosystems. We've been able to take advantage of the fact that individual dolphins can be recognized from patterns of nicks and notches on their dorsal fins, and that allows us to track them through decades. We conduct monthly photographic identification surveys to be able to locate where the animals are, what habitat they're using, who they're with, what their condition is, and their reproductive success. We supplement this with occasional capture-release veterinary health assessments of the animals, where we capture a small subsample of the population, do full veterinary workups, and collect samples from them, and try to keep tabs on the status of the population in that way. We also try to put the animals into an ecological perspective, and we do this through seasonal purse seining of Sarasota Bay in order to be able to look at the relative abundance of the primary prey species of these animals. You put all these pieces together, and what you get is a unique natural laboratory situation for dolphin research and population monitoring. It's unprecedented anywhere in the world in terms of the level of information and the time frame over which the information has been collected. We found that there's a multi-decadal, multi-generational, year-round resident community of about 170 dolphins that uses Sarasota Bay. Within this population, we have up to 5 concurrent generations of individuals, and we have animals that are up to 67 years of age. These animals inhabit a home range that goes from the southern edge of Tampa Bay, Tarasea Bay, and the Manatee River southward to Venice Inlet, and offshore to about 1/2 mile. Over the decades that we've been working with these animals, we've been able to identify shifts in population parameters and ranging patterns that have followed dramatic changes in prey fish availability, for example, as one example of an environmental disturbance. The most impressive example we had of this was the 2005-2006 red tide, during which time more than 90% of some of the primary prey fish of the dolphins were lost to that red tide. And then at a somewhat lower level, the 2010 cold event, which also resulted in extreme fish kills. Tracking these over time, we find that the dolphins' population parameters shifted with the decline in prey availability. During those 2 events, we had declines in population abundance and also in calf production. Fortunately, over the last several years, we've seen an increase in the availability of prey in Sarasota Bay and a concomitant increase in the number of calves born each year and in the number of individuals using the bay. So Sarasota Bay's long-term resident bottlenose dolphins are well suited to serve as indicators of ecosystem health. They show strong site fidelity to a well-defined home range, even when faced with dramatic environmental changes, so monitoring can potentially detect changes in a variety of measurable parameters. We have well-established long-term baseline data on these animals and the prey fish that they consume. Continuous, consistent data collection increases the sensitivities for detection of changes such as those associated with climate change. I've presented examples today from fairly extreme environmental changes. We've also been able to detect finer scale changes, for example, changes in nitrogen levels in the environment, in the tissues of the dolphins that allow us to use these animals as sentinels of what's going on out in the environment. And I'd like to very much thank the Charles and Marjorie Barancik Foundation and the Elizabeth Moore Foundation for helping make it possible for us to continue this long-term research. Thank you.